Welcome back, everyone, for another deep dive. This time we're looking into chromosomal mutations.
Oh, yeah.
Really interesting stuff. So we've got a textbook chapter here that's going to be guiding our conversation.
Okay.
And it's pretty wild to see just how impactful these mutations can be. I mean, we're talking like the evolution of plants and animals,
Wow.
human health conditions,
Ah.
even with like new agricultural hybrids,
Right? Yeah.
those seedless watermelons that you
Yeah.
love.
Oh, yeah. Those are great.
That's
Yeah.
that's kind of chromosomal mutations at work.
Yeah, and the disease-resistant crops and things like that.
Exactly.
It's
So it's
amazing.
like these tiny changes at the chromosome level
Yeah.
that are kind of driving
Huge changes. Yeah.
huge changes. Yeah.
Yeah, it's fascinating how just, a little variation
Yeah.
in the number or the arrangement of chromosomes can have such a big impact on an organism.
Totally.
Yeah.
So let's jump right in. The chapter breaks down these variations into two main types,
Yeah.
aneuploidy and euploidy. Both of them deal with an abnormal number of chromosomes, but in kind of different ways.
Right. So with aneuploidy, you're talking about like a gain or loss of a single chromosome. So it's not a full set. It's
Yeah.
just one chromosome, too many or too few. And then aploidy is when you have the gain or loss of a whole set.
Okay. So like with a human cell normally having 46 chromosomes, would euploidy mean like having 69 or 23?
Exactly. Yeah, you're adding or subtracting complete sets. So the textbook gives us some specific terms to use for these variations. So like monosomy means you're missing one chromosome. Trisomy means there's an extra one. And then for euploidy, we've got terms like triploid and tetraploid. Triploid meaning 3 sets and tetraploid meaning 4.
Gotcha. That makes sense. So I'm curious, though, like how plants and animals handle these variations differently. Because the chapter mentions that plants are way more tolerant of these abnormal numbers.
Mm-hmm.
Why is that?
Yeah, that's a great question. And I think it has to do with their immobility and how they've evolved. So, plants can't just up and migrate if their environment becomes unfavorable.
Yeah, that's true.
But in some instances, these extra copies of genes that they get can actually be beneficial. Like it can lead to larger fruits or a greater tolerance to, say, drought or salt or, something like that. So it's like they have, I don't know.
More tools in the
Yeah,
toolbox.
tools in their toolbox to help them survive, especially when their environment keeps throwing curveballs at them.
Yeah, it's like a built-in genetic backup plan for those unexpected
Mhm.
situations.
Exactly.
But before we like get too deep into the polyploidy side of things, I kind of want to understand how these variations happen in the first place.
Right.
The chapter points to non-disjunction during meiosis
Oh, yes.
as like the main culprit.
Yeah. So non-disjunction during meiosis. Meiosis is the process of creating those sex cells, the sperm and the egg.
Right.
And it's the process of cell division that reduces the number of chromosomes in half. But sometimes those chromosomes don't separate correctly during that division. And so you end up with a sperm or egg cell that has an extra chromosome or is missing one. And then if one of those gametes ends up being involved in fertilization, well, then that variation gets passed on to the offspring.
Oh, wow. So it's like a cellular game of musical chairs, and sometimes a chromosome gets left without a seat.
Yes, I like that. That's a good analogy.
So the chapter focuses on the effects of aneuploidy in humans. And one striking thing that it points out is that monosomy, the condition of missing a chromosome, is usually lethal,
Right.
with one exception, Turner syndrome.
Yes.
So why is that? Why are most monosomies lethal?
Well, I think it really disrupts the delicate balance of genetic information. Each gene plays a role in development. And so if you're missing an entire chromosome, it causes like a cascade of developmental problems. Turner syndrome, which occurs when a person has just one X chromosome instead of the usual 2 sex chromosomes. That's a rare exception. But even that comes with, a bunch of health challenges.
Yeah. And the chapter mentions that with Turner syndrome, the non-disjunction event usually originates from the mother.
Right.
Is there a reason for that?
Well, we don't have all the answers, but it does suggest there's a difference in how sperm cells and egg cells are formed and how they're maintained. So, I mean, think about it. A female's egg cells, they actually begin to develop even before she's born. And then they stay in like this state of suspended development for years. And so, the machinery involved in making sure those chromosomes separate correctly during this long waiting period, it might be more prone to errors.
It's a long time for those chromosomes to just be like, all right, we got to divide perfectly whenever the time comes.
Exactly.
So the chapter also talks about trisomy. So that's where there's an extra chromosome.
Right.
And Tika mentions 2 conditions, Patau syndrome and Edwards syndrome.
Mm-hmm.
Both of them are associated with very severe consequences.
Right.
So why is that? Why would individuals with Down syndrome, which is also a trisomy, often live past their first year, while individuals with Patau or Edwards syndrome usually don't?
Well, that's a complex question. Researchers are still trying to understand it fully, but it likely involves the specific genes on those chromosomes and, how sensitive they are to having 3 copies instead of 2. Each chromosome carries hundreds of genes, each with a very specific function.
So it's not just about having the extra chromosome, but about which chromosome has the extra copy.
Right.
Gotcha. So speaking of Down syndrome, the chapter mentions that scientists have zeroed in on a region of chromosome 21 called the Down syndrome critical region, or DSCR.
Yeah.
What's so important about that region?
So the DSCR, it contains genes that are believed to be particularly sensitive to dosage. So having 3 copies of these genes, as is seen in Down syndrome, can lead to the, characteristic features of the condition. So things like the distinctive facial features and intellectual disabilities. And so researchers are using mouse models to try to better understand how those extra copies of genes in the DSCR affect development.
So they're like little living laboratories helping us untangle
Yes.
all of that complexity.
Exactly.
That's remarkable.
It is, yeah.
So like with other trisomies, most cases of Down syndrome result from non-disjunction during meiosis.
Right.
But the chapter points out that most of those errors happen during meiosis I. What's going on in that phase?
So meiosis I is the first of those 2 cell divisions where the homologous chromosomes, one from each parent, pair up,
Okay.
and then they separate into different daughter cells. And it's during that separation process that things can go wrong. And interestingly, maternal age plays a significant role.
So like the older the mother, the higher the chance of Down syndrome?
Yeah, that's the prevailing theory. The longer those egg cells are arrested in meiosis I, the higher the chance of those errors during that chromosome separation.
It's kind of like a delicate dance. And with age, a few of the steps get messed up.
Yeah, that's a good way to put it.
So this whole thing about maternal age has led to a lot more awareness about genetic counseling for older mothers so they can make informed decisions.
Right.
It's just incredible how these like tiny cellular events can have such big impact.
Yeah,
The
absolutely.
chapter also talks about familial Down syndrome, which is different from those typical cases caused by random non-disjunction events.
Yes. So familial Down syndrome is actually caused by a chromosomal rearrangement called a translocation. Specifically, a piece of chromosome 21 gets attached to another chromosome, usually chromosome 14.
Okay.
Now, someone can carry this translocation and be phenotypically normal. They still have 2 copies of all the essential genetic material.
They just have it.
Yeah, they're just rearranged a little bit.
Rearranged a little bit.
But during reproduction, this translocation can lead to a gamete that has an extra part of chromosome 21, and so that can lead to Down syndrome in the offspring.
So even though the parent doesn't have Down syndrome, they can still pass on this rearranged chromosome
Yeah.
and increase the chances of their child having the condition.
Right. Wow, it's complicated.
It's complicated.
Yeah.
That just highlights how these changes can be passed down through generations. And before we move on from aneuploidy, I did want to touch on this point that the chapter made that a lot of pregnancies involving aneuploidy don't even make it to term.
Oh, that's right. A very significant percentage of miscarriages are due to these chromosomal imbalances. So that emphasizes just how finely tuned that balance needs to be for embryonic development.
Wow. So while aneuploidy can lead to some very serious conditions in humans, plants, as we were saying before, are a different story.
It's totally different.
They can handle polyploidy, that condition of having extra chromosome sets, like it's nothing.
Like a pro.
Yeah. In fact, it's incredibly common in the plant world.
It is, yeah.
So when we come back, we'll dive into that fascinating world of polyploidy and figure out why plants seem to love those extra chromosomes so much.
Sounds good.
All right, we'll be right back after a short break.
Okay.
And we're back. We were just talking about how plants handle polyploidy, having extra sets of chromosomes, so much better than animals do.
Yeah, like way better.
I mean, for humans, even one extra chromosome can be a big problem, but some plants are out here thriving with 3 or 4 or even 8 sets. How do they manage that?
Well, plants have a few tricks up their sleeve. I guess I should say up their stem.
Yeah.
So first off, a lot of plants can reproduce asexually, meaning they can essentially just clone themselves, and that lets them bypass all of the potential issues that come with trying to pair up chromosomes during meiosis when the numbers are off.
So they can just skip that whole chromosome pairing problem altogether.
Exactly.
Clever. But the textbook also describes 2 main types of polyploidy, autopolyploidy and allopolyploidy.
Right.
What's the difference between those?
So autopolyploidy, you can think of that as like a doubling or even tripling of a plant's own genome. So instead of the usual two sets of chromosomes, an autopolyploid might have four or six sets, all from the same species.
So it's like hitting copy paste on their own genetic material.
Yeah, exactly.
But how does that lead to those interesting traits that we see in a lot of polyploid plants, like the larger fruits or the seedless
Right.
varieties?
Well, that's where gene expression comes in. So having those extra copies of genes can change how those genes are actually expressed, almost like turning up the volume on certain traits.
Okay.
And this can result in, larger plants, more vibrant flowers, or even those seedless watermelons that we talked about earlier. All of these changes can be very advantageous for the plant, especially when you consider that they can't move around to escape tough conditions.
Right. Yeah.
So it's like they've got to adapt genetically.
So like those massive pumpkins that we see at the pumpkin patch in the fall.
Mm,
Could that be autopolyploidy at work?
it very well could be, yeah. And those seedless grapes that you enjoy snacking on,
So good.
those are often the result of autopolyploidy as well. In fact, the textbook mentions that commercial bananas are triploid and certain varieties of apples and potatoes are tetraploid.
Wow. It's kind of crazy to think that like a lot of the fruits and vegetables that we eat
Yeah.
owe their existence to this kind of like genetic work.
Yeah, it really is. It makes you appreciate the power of, these genetic variations.
For sure. But autopolyploidy isn't the only way that plants get these extra chromosomes. What about allopolyploidy? The chapter mentions that it involves hybridization, which sounds kind of complicated.
It is a bit more complicated. So allopolyploidy happens when two different plant species hybridize, meaning that they combine their chromosome sets.
Okay.
Now, usually the first generation hybrid is sterile because those chromosomes from the different species have trouble pairing up during meiosis.
Hmm.
However, if that hybrid's entire genome doubles, then you get a fertile allopolyploid.
Wait, so two plants from different species make a baby plant, and that baby plant can't reproduce, but then its genome doubles, and suddenly it can have babies again.
That's exactly what happens.
Wow, that is amazing.
It really is. The textbook highlights how American cotton is a naturally occurring allopolyploid. It's got 26 pairs of chromosomes, 13 large and 13 small. And scientists, they kind of put the pieces together and realized that this likely arose from a hybrid between an old world cotton species, which had those 13 large chromosomes, and a wild American cotton species with the 13 small ones.
Okay.
And they even went so far as to recreate this hybridization in the lab, and it worked. So they basically prove that American cotton's origin story is rooted in allopolyploidy.
Oh, wow, that's amazing. So it's like a genetic detective story, and they figured out how this cotton came to be.
Exactly.
But not all of these hybrids are as successful, right? The textbook mentions this radish cabbage hybrid.
Oh, yes, the radish cabbage hybrid. That one was kind of a flop.
Yeah.
So the idea was that you would combine the, tasty root of the radish with the nutritious leaves of the cabbage.
Okay.
So they did create some fertile hybrids, but the results weren't that appetizing. The root ended up being more like a cabbage, and the shoot was more like a radish.
A cabbage-like radish. I don't know if I'd want to eat that.
Yeah, I don't think it would have been a hit at the farmer's market.
Probably not. Thankfully, the chapter also mentions a more successful hybrid, triticale, a cross between wheat and rye.
Ah, triticale. That's a great example of how allopolyploidy can actually lead to valuable agricultural crops. So wheat has a high yield, but it's susceptible to various diseases. Rye, on the other hand, is more tolerant to harsh conditions, but it has a lower yield.
So the scientists decided to kind of combine their strengths.
Yeah, exactly. By crossing a tetraploid wheat with diploid rye, and then they induced chromosome doubling, they created a hexaploid triticale. And this triticale, it combines the high yield of wheat with the hardiness of rye, and it even has a higher protein content than either parent.
Oh, wow. That's like a super grain.
Yeah, it's pretty impressive.
So we've seen how both autopolyploidy and allopolyploidy can be beneficial for plants.
Right.
But the textbook also mentions one more type, endopolyploidy. What is that?
So endopolyploidy is a little different. It's when only certain cells within an organism become polyploid, but not the whole organism. It's like some cells decide to bulk up their DNA.
Do these cells have a reason for doing that? Like, why would only some cells become polyploid?
Often it happens in cells that need to produce large amounts of certain proteins.
Okay.
So for example, some human liver cells, they can have 4, 8, or even 16 times the usual number of chromosomes, and this helps them to really ramp up that protein synthesis.
Oh, wow. It's like they're specialized protein factories.
Yeah, exactly. And the textbook actually gives a really extreme example, the water strider, Geras. Its salivary gland cells can have over 1,000 copies of each chromosome.
1,000. That's wild. I wonder why they need so many.
That is a mystery.
Yeah.
But it just goes to show how adaptable and diverse polyploidy can be in nature.
So we've explored polyploidy and seen how it can benefit plants. But the textbook also points out that chromosome mutations aren't always about changes in number. There's also rearrangements. So this is where the structure of a chromosome gets mixed up without any loss or gain of genetic material.
Exactly. Those rearrangements, they can be just as impactful as changes in chromosome number.
Wow.
And we'll start by looking at inversions and translocations, two key players in that chromosomal reshuffling.
Okay, let's start with inversions.
Mhm.
The textbook describes them as a segment of a chromosome breaking off, flipping, and reattaching in reverse order. It's kind of like taking a sentence and reversing the order of a few words.
Yeah.
You might still be grammatically correct, but the meaning could be totally different.
That's a great analogy. And you're right, inversions, they can disrupt gene function if they happen to fall, within a gene or its regulatory regions. But sometimes they can be neutral or even beneficial evolution over long periods of time.
Okay, the textbook does mention this beneficial aspect,
Yeah.
but it doesn't really dive deep into it.
Yeah.
Can you kind of elaborate on how an inversion could be beneficial?
Sure. Imagine you have a set of genes on a chromosome and they work really well together. Okay.
Providing some sort of advantage to an organism.
Mm-hmm.
Now, during meiosis, chromosomes exchange segments through crossing over.
Right.
But if an inversion happens, it can essentially lock those beneficial genes together,
Okay.
preventing them from being separated during that crossing over event.
So it's like putting a protective barrier around them so they get passed down as a unit.
Exactly.
That's fascinating.
Yeah. And these inversions, they can help to maintain these beneficial gene combinations,
Okay.
which contributes to a species' adaptation over time.
So they're like the guardians of those good genes.
Yeah, I like that.
What about translocations? The textbook describes them as exchanges of chromosome segments between non-homologous chromosomes.
Right. You can think of it like swapping pieces between different puzzles.
Okay.
The simplest type of translocation, you have two breaks, one on each chromosome, and then those broken pieces swap places.
But even if you're just rearranging the existing pieces, wouldn't that still disrupt the genetic information? It definitely can, especially during meiosis. So when an individual has one normal chromosome and one translocated chromosome, those chromosomes actually have to contort into unusual shapes to pair up properly.
Okay.
And that can lead to the gametes ending up with duplications or deletions of certain chromosome segments, which can cause problems for the offspring.
So even though there's no overall loss or gain of genetic material, those rearranged chromosomes can still cause problems.
Yeah.
And the textbook mentions that this often leads to reduced fertility,
Yeah.
also known as semi-sterility.
Right. It highlights how important the precise arrangement of genes on chromosomes is for normal development.
For sure.
And the chapter actually gives us a great example of this with familial Down syndrome.
Right. We talked earlier about how most cases of Down syndrome are from non-disjunction, but familial Down syndrome is caused by a Robertsonian translocation. So that's where the long arm of chromosome 21 gets attached to another chromosome.
Right. And individuals that carry this translocation are usually phenotypically normal. Okay.
Because they still have two copies of all of the essential genetic material, they're just arranged differently.
Gotcha.
But when they reproduce, there's a chance that a gamete will end up with both the normal chromosome 21 and the chromosome with that attached segment, leading to Down syndrome in the offspring.
It's a reminder that these rearrangements can have effects across generations.
Definitely.
So speaking of chromosome vulnerability, the chapter also talks about fragile sites.
Right.
What are those?
So fragile sites, those are specific regions on chromosomes that appear as gaps or breaks when the cells are grown in the lab without certain chemicals.
Yeah.
So basically, they're like weak points in the chromosome structure.
So like fault lines where earthquakes are more likely to happen.
Yeah, that's a good way to visualize it.
Right.
And these fragile sites, they seem to be more prone to breakage and rearrangement, which can lead to various genetic disorders.
Okay. And the chapter dives into Fragile X syndrome, the most common inherited form of intellectual disability.
Yeah.
What makes that condition so unique?
Right.
Well, Fragile X syndrome, it's caused by a fragile site on the X chromosome, specifically within this gene called FMR1. And this gene, it contains a repeating sequence of 3 nucleotides, CGG.
Okay.
And individuals with the normal number of these repeats are fine, but when that number expands beyond a certain threshold, it disrupts the gene's function, and that's what leads to Fragile X syndrome.
The chapter also mentions this interesting phenomenon called genetic anticipation,
Mm.
where the severity of Fragile X seems to increase in future generations. Why would that be?
Well, that's because the number of those CGG repeats actually tends to increase as the gene is passed down through families. So a parent might have what's called a premutation with a moderate number of repeats, and they might only have mild symptoms or no symptoms.
Yeah,
But then their child could inherit a full mutation with way more repeats, leading to much more severe symptoms.
so it's like the mutation itself is evolving over time.
Yeah, it's a bit unsettling.
Yeah.
Yeah.
And fragile sites aren't only linked to Fragile X syndrome, right? The chapter also mentions a connection to cancer.
Yes. So there's this gene called FHIT,
Okay.
and it's located on a fragile site on chromosome 3, and it's been linked to several different types of cancer. So it seems like genes that are located within these fragile sites, they're more susceptible to damage.
Okay.
And that damage can increase the risk of developing cancer.
It's like those fragile sites are these ticking time bombs within our DNA.
Yeah.
Yeah.
It's a good way to think about it. It's remarkable how these tiny, vulnerable regions can have such a huge impact on our health.
So we've covered a lot of ground today, from how plants can handle all those extra chromosomes to the complexities of these chromosome rearrangements in those fragile sites.
Right.
What are your final thoughts as we kind of wrap up this part of our deep dive?
Well, I think it's just amazing how these seemingly small changes at the chromosomal level can have such far-reaching consequences for individuals and entire species. And as we learn more about these mutations, it opens up so many new avenues for understanding evolution, for developing therapies, for genetic disorders, and even improving agriculture. It's really exciting.
And we're back for our final segment on chromosomal mutations.
Yeah.
So far, we've been focused on the science behind all of this, but obviously there are some bigger implications to think about.
Oh, definitely. The more we learn about genetics, the more ethical questions pop up.
Right, like how should we be using this knowledge?
Exactly.
And what are the potential consequences?
Yeah.
So the textbook brings up Down syndrome as an example.
Yeah.
It's the most common chromosomal abnormality in newborns.
Mm-hmm.
And we've had prenatal diagnostic tests for it for decades.
Right.
But what happens when a fetus tests positive? Like, what kind of decisions do parents face?
Yeah, that's a really tough situation.
Yeah.
There's no easy answer. I mean, the decision to continue or terminate a pregnancy after a Down syndrome diagnosis, it's so personal. It's influenced by so many factors.
Yeah, the chapter doesn't really get into all of those factors, but
Yeah.
it's definitely a complicated decision with a lot of, emotional, ethical, and societal implications.
For sure. And we can't really ignore the historical context of eugenics when we're talking about these things.
Yeah.
I mean, the idea of manipulating human populations to get certain traits has a pretty dark past.
It's a scary thought that scientific knowledge can be twisted and used for harm like that.
Yeah.
The chapter even mentions those forced sterilization programs
Right.
from the early 20th century,
Oh.
which is just horrifying, like how eugenics was used to control reproduction and discriminate.
Yeah, it's a huge warning sign from history.
Yeah.
And it makes these conversations about modern genetic technologies that much more important. We've got to make sure they're being used responsibly and ethically,
Yeah.
with respect for individual choice and human diversity.
So that brings us to this question whether modern practices like prenatal testing and selective abortion are basically a new form of eugenics.
Right.
It's definitely a debate with strong arguments on both sides.
Oh, yeah, for sure. Some people say that these individual choices about reproduction shouldn't be compared to like those state-sponsored eugenics programs.
Right.
Parents have a right to make informed decisions about their families and the challenges they might face.
Right. But other people worry about the potential for discrimination
Yes.
and devaluing people with disabilities.
Absolutely.
They're concerned that by choosing against certain traits, we're saying that those traits are undesirable.
I understand that concern.
Yeah.
Yeah, it's a really nuanced conversation.
For sure.
And I think we need to be careful about balancing individual reproductive freedom with societal values
Right.
and make sure that our choices aren't reinforcing harmful stereotypes.
Yeah. It's a lot to think about.
It is.
No easy answers. So the textbook moves on to a different but kind of related topic.
Yeah.
Using triploid grass carp to control invasive aquatic vegetation.
Huh.
What's that all about?
So this case study, it shows how we can use genetics to solve real-world problems. But like you said, it comes with its own ethical and ecological questions.
So how are these triploid carp different from like regular carp?
So triploid carp are bred to have 3 sets of chromosomes, which makes them sterile.
Okay.
And that sterility is important because it means they can't reproduce uncontrollably and become invasive themselves.
Okay.
They're put into waterways to eat invasive plants.
Oh, so they're like little swimming weed whackers?
Exactly. And because they're sterile, we don't have to worry about them taking over.
That does sound like a pretty elegant solution.
It does, but there are potential downsides.
Like what?
Well, they might eat native plants along with the invasive ones,
Oh, right.
which would disrupt the ecosystem.
Yeah.
They could harm other organisms that depend on those native plants.
So even with good intentions, there can be unintended consequences.
Exactly. We have to be really careful when we're messing with complex ecosystems.
Well, looks like we've reached the end of our deep dive into chromosomal mutations.
Wow, that went by fast.
I know. We've learned so much from the science behind these variations to their effects on plants and animals, and even those ethical questions they bring up.
It's been fascinating.
So just to recap, we talked about how delicate the balance of chromosomes is, and how even small changes can have big impacts. And as we've seen, this knowledge can be used in many ways, from agriculture to medicine.
Absolutely.
And who knows what new discoveries and challenges are waiting for us in the future.
I'm excited to find out.
Thanks for joining us on this deep dive into the world of chromosomal mutations. We hope you enjoyed it and learned something new.